All right, let's test the microphone here. See, I almost had the wrong one selected again. That's going to be great. All right, hope everyone's doing well. Welcome to back to Biotechnology, Saturday night biotech. Awesome. All right, so this is a important week for you guys. You have an exam coming up. We get over here and just revisit your requirements for this week. So the exam is going to go live some Sunday night at a minute after midnight. It will become available. You have a week to complete it. Where's my schedule here? The exam will cover chapters 4 through 7. All right, so the exam is a little shorter than usual. It's 35 to 33 questions. So I'm going to tell you most of the questions are from chapters 4 and 5. There's only about 4 questions from tonight's lecture on animal biotech and about an equal number of questions from plant biotech. Okay, so remember, exams in this course use respondus, right? They're closed book. You guys are expected to study for the exam and take the exam in a honest environment. And don't forget, I do check these videos, not in a creepy way or anything, but I do scan them, and they give me flags for potential acts of academic dishonesty. Okay, so in my experience, it's usually around exam 2 or 3 when we start getting issues with this, and especially this being a 2000 level course. I know a lot of you guys, most all of you, should be freshmen and sophomore, and you don't want to deal with those problems, right? They're just a giant headache, not only for me, but most especially for you. So please just, take the exam like you're supposed to take it. All right. Okay, that's all I want to say about that. You guys are a good group. I'm not exceedingly worried, but like I said, it's usually exam 2 and exam 3 in the online courses where these problems start to come out. So I hope they don't. I don't like those problems. They're very, very icky. Okay, so you have a week to complete that exam. I'm of course posting the final lecture and the content for that exam tonight, which is on chapter 7. And then next week I'll post chapter 8. That's on exam 3. Only 3 chapters. 3 chapters on exam 3. But so the reason I did that, that you only have 3 chapters on exam 3, and why I will give you a little hint that this exam 2 is not exceedingly difficult. So it is an opportunity for all of you to get 100. I mean that. And it's because I don't want you to kill it yourselves. I don't want you guys killing yourselves too much on this week. I really would prefer that you guys are putting in some work in your research paper right now. So, keep that in mind. I don't blow this exam off, but it is a really good chance for you to get a very high grade. Okay, so that being said, I did provide a review sheet. So if you go to course content, you'll see a little exam review sheets there. And I have the review sheet right here. Just like the last review sheet, okay, take this for the value that it is. I basically went through the questions and kind of summarized the topic. So between this and your PowerPoint slides and your textbook, I think you will be very well prepared for your exam. So best of luck. Reach out if you have any issues or problems. And remember, it is due by Sunday at midnight. Not tomorrow, a week from next weekend. It's due. Okay. All right. This is not going to be an exceptionally long lecture tonight. because I did remove quite a bit of material. We are going to cover this chapter in animal biotechnology. And I like to have this thing open like this, but I know you guys probably prefer it larger on the screen. Let me see if I can minimize that. I can. Good. Okay. So, animal biotech. So, you can see right there, section 7.2 on cloning. I took that out. I really didn't like it. I didn't like how the author did it. So, we'll talk about Dolly later on. Dolly was a sheep that was cloned, the first animal ever cloned. We'll talk about that later on in another chapter again. So, I'll just bring it up then. So, you just have sections 7.1, 7.3, and 7.4 on this exam. All right. So, genetically engineered animals. All right. So there's a lot of weird terms for this stuff: genetically engineered, transgenic, animal biotech. Trying to draw lines around it can be difficult. They really blend together a lot. And there's a lot of truth in that. They're all kind of the same thing. I prefer the term genetically engineered or transgenic. And these are animals that have either been bred for a particular phenotype, just like we saw in plant biotechs, or they have been genetically enhanced or genetically changed. So either through mutation or through CRISPR or pronuclear injection, all these different methods are available to genetically engineer animals. So what sort of benefits do they provide? Well, when we're trying to study medicine, right? I mean, you can only get so far in studying a biology like that in a plant, right? So you need you need a living system that is closer to a human. And we're going to go over some different model organisms tonight. But that is not the only reason for doing or for generating genetically engineered animals. So medical treatments, of course, is definitely makes a hotter topic. It's a little sexier of a topic, but improving food and also veterinary science, welfare of animals. I was watching the news yesterday, and it looks like the FDA has approved a drug for canines that increases lifespan, which is, to get FDA approval on something like that is it's pretty remarkable. I haven't looked at any more depth than that, but you guys are welcome to do that. I don't know the name of the drug or what the mechanism of the drug is, but the fact that they got FDA approval is pretty remarkable. So obviously there's a lot of ethics involved in this. I'm an animal lover, and although I don't have any right now, I've got a fish tank. It's about as crazy as it gets over here. I've got 3 kids. Those are my animals. So, all right. So, just some examples that the text provides on how animal research, how important it is. The polio vaccine, it was originally generated using animals. Nowadays, we generate vaccines using cell culture, but vaccines originally generated animals, and the millions of lives that have been saved from vaccinating against polio is pretty well accepted. Mechanisms, so treatments like dialysis, heart surgeries, whenever something gets approved by either a medical board, the FDA, it usually has to first go through some sort of animal test, right? So, dialysis, that whole working all that out with the membranes and pulling the blood out of someone, running it through the machine, all the various filters and membranes, that all had to be optimized in an animal. The other example they gave was cataract surgery, right? So that had to be optimized in an animal as well. So almost like a side effect, though, of a lot of the human health discoveries and progress that's been made in animals has been the development of veterinary biologicals and vaccines as well. Obviously, we're trying to generate a avian flu vaccine right now that's useful in birds. What are some example model organisms? So, most people think of the mouse, and the mouse is a wonderful model organism, like enormous genetic homology between a mouse and human. But for certain studies, rats might be better. Remember, a rat's a little larger if you have to do any sort of surgery or whatnot. It's a little easier to perform in a rat. Rats might provide better answers in terms of toxicology than mice. But so at a, if you're doing research at a university, there is a whole board. It's called IACUC, and they evaluate, your need to use animals in research. And I can tell you, it's a very, very difficult process. There's a lot of scrutiny, a lot of steps. It doesn't happen overnight. It usually takes about 1 to 2 years just to get approval to use a mouse in a study. These are mammals, these are vertebrates, and this isn't like Frankenstein medicine we're talking about here. And actually, the numbers of animal experiments have been declining over the last 20 years pretty significantly. But there's also other model organisms that are very useful as well. The fruit fly, Drosophila, is a wonderful model organism for studying genetics. All you need is a little rinky-dink microscope, and you can see a lot of the phenotypes that are available on the fruit fly. Nematodes are another great genetic model. We can easily turn genes off in the nematode, so very nice model to use if you want to target specific genes and see what sort of effects they have. Also, with